Welcome back to PLS 510. Today we're going to take up the topic of forage anti-quality factors and disorders. The first two that we will take up are nitrate toxicity and prussic acid poisoning. But first I'd like to tie this lecture back to the summer annual grass lecture that precedes it by pointing you to this table, which happens to be in the latter part at the end of our publication, AGR 229, the overview publication on warm season annual grasses for Kentucky. This is a table of characteristics of our commonly used summer annual grasses. You'll notice the species on the left-hand side, from crabgrass all the way down to Sudan grass, and the characteristics along the top. Now, of course, today we're going to focus a lot on nitrate toxicity potential and the prussic acid potential. But you can also see some of the other characteristics such as suitability for silage or hay or grazing for all of these species. So let's move on. There are several forage disorders that are due to anti-quality factors, and we're going to list a few here. The ones we will take up today are the top four: nitrate toxicity, cyanide poisoning, bloat, and grass tetany. In a later lecture, we will look at botulism and listeriosis. I want to start right out with this table, or this pair of tables, comparing nitrate toxicity on the left and cyanide poisoning on the right, because these two are often confused, and we'll talk about that or in comparison quite a bit here in the next few minutes. As I said, they're often confused, and let's look at why. Nitrate is highest in the lower stems. Cyanide, on the other hand, is highest in the leaves and the young tissue. Nitrate is lowest in leaves and seed. Cyanide is lowest in the stem, so it's exactly backwards. Nitrate is not metabolized by the drying of hay. Think of it as salt. And if you take up salt into a fresh green leaf and then dry that leaf, it doesn't get less salty. It will, if you're tasting it, it's going to appear more salty. So that nitrate remains and the water leaves. In the case of cyanide, after hay cutting, dissipates as a gas. So the drier the hay, the lower the cyanide. And similarly for the two, ensiling reduces the toxicity significantly for both nitrate and cyanide. So let's look at nitrate accumulation and toxicity just a little closer. In order to get nitrate toxicity, we have to have a combination of things. Number one, we need to have high nitrogen rates, and we need adverse conditions that prevent the plant from metabolizing the nitrates that it brings up through the roots and into protein. The things that will cause, what are those adverse conditions? They could be drought or a sudden change in the weather. Since we're talking about summer annuals here, usually it's changing from very warm and clear to cloudy and cool, and all of a sudden the growth stops, or the growth is not very fast, and nitrates are not metabolized. So in those cases, nitrate will accumulate in the lower stalks and stems. And again, leaves and seed are low. Nitrate persists in hay and metabolized in silage. Nitrate toxicity can occur in all animals, but ruminants are much more sensitive, and here's why. The nitrate that's present in plants, once it gets to the rumen, it's converted to nitrite. That nitrite will bind with hemoglobin to make methemoglobin or metallohemoglobin. And what that really is the iron molecule that's in the middle of every hemoglobin molecule that is able to transport oxygen. That iron molecule is in the +2 or ferrous state, if you can think back to your inorganic chemistry. When nitrite binds with hemoglobin to make methemoglobin, it flips that nitrogen to +3 state, or the ferric state, and in this state it cannot transport oxygen, and so animals die of asphyxiation, and rather quickly too. Horses can be affected, as can most monogastrics, but the thresholds are not clear. Our most common nitrate sources are drought-stressed corn or sorghum species, but I don't want you to ever forget about Johnson grass, because it can be a source of both nitrates and prussic acid. Brassicas can also be a source, as can the weeds ragweed, pigweed, thistle, and dock. You will also notice and be relieved that these are weeds that livestock don't readily consume. Nitrates can also enter the diet of a ruminant through the water sources through contaminated wells. I wanted to include this table mainly for a couple of reasons. First of all, if you need to find a reference for toxicity and you have a test for nitrate, you can look at the table at the top of this page. And this table is found in your pocket guide, it's also found in Southern Forages. In our case, we also reproduce the table in ID 217 on nitrate poisoning. But you may be called upon to convert different forms of nitrogen, such as the nitrate nitrogen here in the second row of the bottom table, to nitrate, just plain nitrate. In that case, you would multiply by 4.4. And so that table is useful. You need to know where it is and know that you may be called on when you're practicing to be able to interconvert forms of nitrate. I'd like to switch gears now to prussic acid poisoning. Prussic acid is cyanide, HCN, or hydrogen cyanide. It gets its name because cyanide was isolated from a dye called Prussian blue, hence prussic acid. Sorghums contain durin, which are cyanogenic, not glucosides, but glycosides within the epidermal cells of the leaf, and this significance will become clear in just a moment. Enzymes that are responsible for hydrolyzing durin into two parts, the sugar part and the cyanide part, are found within the mesophyll cells of the leaf. So an ordinary intact leaf has the potential to make prussic acid, but it is not going to do so as long as it is intact. When we chew those leaves or masticate them, it brings the enzymes into contact with durin and it releases cyanide. Like nitrate poisoning, the rumen environment can make this worse. It does this by acting directly on the durin to also release cyanide, so we can get cyanide from the chewed up tissue, but whatever might not have been mixed and start to evolve cyanide whenever it's chewed up, when it gets to the rumen, it can be durin can be further acted on to release cyanide. Cyanide inhibits cytochrome C oxidase within the electron transport chain of the mitochondria and causes death by asphyxiation very quickly. Let's take a look at some species that we're going to be careful with or look to avoid or prevent cyanide toxicity. We've mentioned the forage sorghums, and that's sorghum Sudans, and Sudan grasses, as well as their perennial cousin, Johnson grass. Wild cherry leaves are also a potential. Indian grass might not be on your list, but if you think back to the scientific name for Indian grass, that was Sorghastrum nutans. So it has prussic acid potential. And white clover, really, white clover. This became brought home to me during the mare reproductive loss syndrome when we were trying to figure out what is it in the environment that's insulting those pregnant mares and causing them to abort. And if you start taking a look at all the toxic plants that might be in horse pasture, you've got to ask, or you got to, well, you can easily find that white clover is known to, certain varieties and certain types are known to have a fair amount, not as much as some other plants, but a fair amount of prussic acid potential. And as we stated earlier, horses may be sensitive to prussic acid, but the thresholds are not very clear. In fact, we did wind up doing some experiments dosing horses with cyanide, and it caused them to be uncomfortable, it caused them to sweat, and it caused them to do a lot of things, but it didn't cause them to abort. I wanted to include this to show you visually that separation of durin and the enzymes that will hydrolyze it. So the epidermis, or that outside layer of cells, contain durin. The mesophyll, or the cells in the middle of the leaf, contain durinase and alpha hydroxynitrilase that are enzymes that will hydrolyze durin, and that is, it'll split it into the cyanide and to the plus the sugar part. How can we prevent cyanide toxicity? Don't graze sorghums until they reach 18 to 24 inches tall. Remember, it's the young leaves and the young green tissue that are highest in prussic acid potential. And of course, plant something else. Pearl millet is similar in growth habit to the sorghum Sudans and Sudan grasses, but has absolutely no prussic acid potential. And for that reason, a lot of producers will choose to plant pearl millet instead of the sorghums. Nitrogen makes it worse, so use moderate amounts of N. And by moderate, I would say that we would use 60 pounds of N in advance of any growth cycle. The way that we manage these tall-growing sorghum Sudans and Sudan grasses and pearl millet is to apply some nitrogen after we graze or towards the end of a grazing cycle, and then pull the animals off and let it come back. And typically we use about 60 pounds of N per cycle every time we want to get regrowth. And if we don't want regrowth, we stop adding in. Another thing that you want to have on your list in order to prevent cyanide toxicity is to not graze cyanogenic plants when frost is likely. And every year this question comes up, and the only correct answer, the only safe answer, is if you expect temperatures to be at or below freezing and you have cattle on Sudan grass or Johnson grass, they need to come off. After the plant has been frosted and killed back to the ground, typically about 2 weeks, then it's safe to turn back in. Please watch the young tissues that come up from the base, because these are going to be very succulent, very attractive that time of year to grazing animals, and they're going to be very high in cyanide potential. And last, of course, watch for fallen limbs of cherry trees after storms. And while we said before that white clover, academically speaking, can have some cyanide potential, it is not of a practical concern for grazing animals. All right. This brings us to a close of this short segment on nitrate toxicity and prussic acid poisoning. Until next time, we will see you then.